Welcome to part 5 of this video on the immune system. Okay, so the big topic that we're going to begin in this video is the topic of the complement system. So to remind you, we are trying to understand what is the point of forming an inflammatory exudate now. We've seen that this is one of the major things that is formed by the acute inflammatory response, but what's the point of it? Well, here is the answer. In the bloodstream, there are loads of proteins which are capable of attacking and killing pathogens. A big group of these are known as the complement proteins, and these are the ones that we're going to study now in detail. So we're going to discuss the different complement pathways that exist for actually killing bacteria, and this will now allow us to understand why we actually wanted to form this inflammatory exudate and why it's going to help clear the infection that is present within our tissue. Okay, so we've said already that the complement proteins are all circulating in the bloodstream in an inactive form, and that they're put into the bloodstream by the liver. What I now want to actually discuss is the 3 pathways that exist for activating these complement proteins and for actually using them to kill pathogens. Okay, so there are 3 different pathways by which the presence of a pathogen can activate the complement proteins and therefore lead to the complement proteins killing the pathogen. And these are known as the classical complement pathway. Then we'll have the alternative complement pathway, and then finally what is known as the lectin complement pathway. And we're going to discuss the the mechanism of each of these 3 pathways in detail. And we're going to begin with the classical complement pathway because it's the one that was classically discovered, and then the others were discovered later. So it's a nice place to start. The problem with starting with the classical complement pathway is that the classical complement pathway won't actually be activated until you've had an adaptive immune response. The classical complement pathway requires molecules which will be synthesized by the adaptive immune response in order to work. So what I'm just going to have to do is briefly introduce you to the concept of an antibody. We will discuss this concept in so much detail later on, but for now I'm just going to give you a brief introduction to the antibodies so that we can then discuss the classical complement pathway. So we're beginning with this one, then we'll talk about the alternative complement pathway, and then the lectin pathway. Okay, so let's draw our little pathogen here. So let's say this is our pathogen, and let's say that in the fluid surrounding the pathogen we now have a huge number of these complement proteins that have come in with the inflammatory exudate. So here's our pathogen. So in the classical complement pathway, what has to happen is antibody molecules are going to bind to the pathogen. They're going to bind to molecules on the surface of the pathogen, and then the bound antibody molecules are then going to activate the complement system. So let me introduce you to what an antibody is. So antibody molecules are molecules that are produced by the adaptive immune system, and they target proteins or molecules, mainly proteins, that are associated with your pathogen. So in an adaptive immune response you produce antibody molecules that will specifically bind to proteins on the surface or which are secreted by the specific pathogen. And for different pathogens you'll have very different molecules, and you'll produce different antibodies which are capable of binding to those different molecules. So that's what I meant when I said the adaptive immune response is extremely specific to the pathogen. This is one example of that you produce antibody molecules which are tailored to be able to bind to proteins that are associated with that pathogen or other molecules. So, the protein could have all sorts of carbohydrate molecules and lipid molecules attached to it. The antibody molecule will be able to attach to that structure, but generally it does have a protein core that the antibody is launched against. Okay, right. So let me just draw one of these proteins that we will say the antibody molecule has been directed against. And such a molecule mainly protein, but as I said, it could have lipid or carbohydrate modifications to it, that is capable of having an antibody produced against it, is known as an antigen. So look at the word here, gen, we've seen that before, to create. Anti refers to antibody, so this is a molecule that is capable of having an antibody created against it. So we call, we have this umbrella term, antigen, like we have the umbrella term pathogen, for a molecule that will generally be a protein, potentially with lipid or carbohydrate modifications to it, which can have an antibody produced against it. And the pathogen could have loads of different antigens on its surface. So now let me show this antibody that has been produced by the adaptive immune system, which is going to bind to this antigen. And as I say, we'll discuss antibodies in a lot more detail later on. So for now, I'm just going to draw a picture of what an antibody looks like. Don't worry about the structure yet. We'll go through the structure in much more detail later on. So this is the picture that at the moment I'm going to draw for what an antibody looks like. This Y-shaped structure here. And you can see that here it is binding to the antigen. And I'll just draw a few little connections, which are actually disulfide bonds. So actually I'll tell you a little bit about antibodies here. They're made up of 4 separate polypeptides, so 1, 2, 3, 4. And I've now drawn these lines which are showing connections between these 4 polypeptides, and these are disulfide bonds. One between this polypeptide here and this polypeptide here, 2 between the 2 central ones here, and then another one on this side between this one and this one. Okay, so just colouring in the different peptides, different polypeptides of the antibody then. Here is one in green, here's an identical polypeptide in green, and here are the other 2 polypeptides, which are again identical. So you have 2 of the two of the pink polypeptides and 2 of the green polypeptides. And the green polypeptides are known as the heavy chains, and the pink polypeptides are known as the light chains. But we'll stop there. We'll go over antibody structure later on. For now, all we need to know is that this molecule is specific against this antigen that's on the surface of the pathogen. It's now bound to it. And having an antibody molecule bound to you like that is a giveaway sign that you are a pathogen, because we shouldn't be producing antibody molecules against proteins on the surface of our own cells. So if you've got an antibody on your surface, and indeed the pathogen will probably end up with loads of antibodies on its surface, that's a giveaway sign that you are a pathogen. And now the classical complement cascade, or the classical complement pathway, depending on what word you want to use to describe it, is going to launch in response to seeing this. This is going to activate the classical complement pathway, and as I said, hopefully you can understand the motivation for that, that if this is present on the surface of something, it's a giveaway sign that it's a pathogen. So we might as well launch this complement cascade to kill it. Okay, so let me now go through the actual classical complement pathway. So what happens first? So the first complement protein that's going to bind to the antibody molecule that's bound to the antigen here on the surface of the pathogen is a complement protein known as C1. And I'll draw this like so. So here is a great big complement protein known as C1, and this is now going to bind to our antigen antibody complex. And I'll colour C1 here in an orange. So C1 comes in and binds to the antibody-antigen complex. And by the way, a complex of an antigen and an antibody is often known as an immune complex. So the C1 is going to bind to the immune complex, as shown here in this picture. And that's now going to activate the C1. And the C1 is now going to activate a whole bunch of other complement proteins. So the 2 complement proteins that C1 is now going to act on are called C2 and C4. So you're starting to get the pattern with how the complement proteins are named. They're all just called C with then a number after it. So C for complement, so this is the complement protein 1, and now we're going to see the complement protein 2 and the complement protein 3. Sorry, not the complement protein 3 yet, the complement protein 4. So here, this is now C2, and let's say this is C4, so these are just present in the inflammatory exudate, but now they're going to be activated. At the moment they're in an inactive form, they're now going to be broken down by the C1, which has been activated by binding to the immune complex. So this is now activated, so I'll draw a few sparks coming from it there. and it's now going to act on the C2 and the C4 here, and it's going to break them down into two separate fragments, one of which is going to be activated. So both of these are going to be broken down by C1, so it will act on both of these, and there'll be loads of these C2 and C4 in the exudate, and it will just continue breaking down C2 proteins and C4 proteins into these fragments. So C2 proteins near the C1 are going to be broken down into two fragments, one small fragment here and one large fragment. Now the small fragments are known as A's, so this is C2A, and the large fragment is known as a B fragment, so this is C2B. And I'll come back and talk in just a moment after I've drawn it out for C4 as well, about a warning here about names for C2's fragments. So now let me talk about C4. So C4, the same thing will happen here. C4 will be broken down into a small fragment and then a large fragment, and the small fragment will be called the C4A and the large fragment will be called the C4B. And I'll colour these two in blue as well here. Okay, so now a warning. So a warning about the naming of the fragments for C2. So when the people who discovered this were naming up the fragments of C2, they had not yet agreed on this nomenclature of A would be the smaller fragments and B would be the larger fragments. They were just using A and B as two letters to label up two fragments. They hadn't agreed that A would be the smaller fragments and B would be the larger fragments. In the modern notation for complement proteins, when we have complement proteins like C2 and C4 which are being broken down into two fragments, we call the small fragment the A fragment and the larger fragment the B fragment. Now, in the old nomenclature for the fragments of C2, when they first discovered the complement pathways and they were naming up the fragments that C2 was split down into, they did it the other way round. They called the small fragment of C2 C2B and the large fragment of C2 C2A. Now this problem didn't actually arise for C4 because they did actually call the small fragments of C4 C4A and the large fragment of C4 C4B. So this naming we have tried to phase it out. It's being phased out, but it hasn't gone yet. Some people will still call the small fragment of C2 C2B and the large fragment of C2 C2A. So you will see this naming for the fragments of C2. It's the old naming, the one that originally was used by the people who discovered the complement pathways. Now what we've tried to move over to is calling all of the small fragments A fragments and the large fragments B fragments. Now this problem doesn't arise for any of the other complement proteins because all of the others, the naming works out perfectly, i.e. according to the names that they gave them years and years ago, the small fragment is the A fragment and the B fragment is the larger fragment. So for all of them apart from C2, the naming was perfectly in accord with what we would use for the naming in this modern era now. I.e. they did happen to name the smaller fragment the A fragment and the larger fragment the B fragment, except for C2 where they got named the smaller fragment the C2B fragment and the larger fragment the C2A fragment. So be aware that if you go to certain sources they will use this nomenclature and that this is a hugely confusing fact. So this is we're trying to phase this out, and this is the one that ideally you would now use for the naming of the C2 fragments. So it is the larger fragments that are actually going to be useful for something. The smaller fragments are now not going to do much. So we're going to continue the story with the larger fragments now, which we will call C2b and C4b, but be aware that some people will call the larger fragments of C2 C2a and would now say that the pathway continues with C2a. Okay, but we will use the modern notation. So, what's going to now happen? So we're generating loads of these fragments now. We're getting loads of C2b produced in the vicinity of this pathogen. We're getting loads of C4b produced in the vicinity of this pathogen. Now C2b and C4b, they are very capable of binding to membranes. Once you've got these B fragments produced like this, they love to bind to cell membranes. So the inactive complement proteins didn't have this desire to bind to cell membranes, but now that we've produced the C2b and the C4b, they love to bind to membranes. So what's now going to happen is, because they're very close to the pathogen, where's the nearest membrane that they can bind to? Well, it will be the pathogen's membrane. So if I now draw the surface of the pathogen enlarged over here, so here's my next picture. Here is the pathogen in red, so this is a bit of the pathogen's membrane. We've zoomed in now. So I've not drawn the entire pathogen, I've just drawn a bit of it. And now what's going to happen is C4b and C2b are going to bind to the cell membrane, and they love to form a complex with one another once they've bound to the membrane. So you end up getting C4b bound to the membrane, binding to C2b, and forming a complex in this way. So let me just colour this in. So here is C4b in blue there, and then here's C2b in red. So this complex here is now called the C4b C2b complex. And again, coming back to my warning, some people would call this the C4b C2a complex. If you see people talking about the C4b C2a complex, and you'll most likely get incredibly confused, you'll be like, did he tell us wrong in that video? Is it C2a that actually binds? No, it's just different nomenclature. It is the big fragment that binds. So C4b C2b complex here. Okay, so you get these C4b C2b complexes forming on the surface of the pathogen then. So what's the next bit? Because these complexes aren't what kill the pathogen, at least not directly. So what are these now going to do? Well, they actually, together, this complex is going to act as an enzyme, and it's going to act as an enzyme capable of activating complement protein 3, C3. So the C4b C2b complex actually functions as what is known as a C3 convertase enzyme, which is just an enzyme which is going to activate C3. So what's now going to happen is these are going to take the complement protein C3, which of course will also be in the inflammatory exudate now. So here is our third complement protein. So we've seen Z1, C2, C3 now, and C4. So what colour shall I go for C3? I think I'll do C3 in green here. So C3 is now going to be broken down into a actually that can be the big fragment now. I've drawn it too big. A big fragment, which is of course called C3b, and a small fragment which is of course called C3a. And again, this is one of the nice ones. There was no problem here because the name that the people who discovered this gave to each of the fragments perfectly fits with our modern nomenclature, i.e. they gave C3b to the big fragment and C3a to the small fragment. Okay, so the C4b C2b complex is going to function as a C3 convertase, and it's going to start converting C3, which is inactive and is just in the inflammatory exudate, into C3b and C3a. And now C3b is going to do the same thing as C2b and C4b wanted to do. It's going to bind to the membrane of the pathogen. It loves to bind to membranes, and the membrane that's closest is the membrane of the pathogen, because remember, this is all occurring right next to the membrane of the pathogen, so likely the C3b is going to end up binding to the pathogen. Okay, and also note that these C4b C2b complexes, they will be catalyzing loads of these reactions. They'll be breaking down absolutely loads of C3 proteins into C3b and C3a fragments, and therefore you'll get loads of C3b proteins being produced in the vicinity of the pathogen, and therefore the pathogen actually is going to end up coated in C3b proteins. So let me just draw this out for you. So here is our pathogen, like so, and now it's going to end up coated with C3b proteins. So all of these are representing C3b proteins, like so, and I'll just colour them all in green. So you end up coating your pathogen in C3b proteins, and these now are actually going to help kill the pathogen, because what does C3b do? C3b is what is known as an opsonin. So we describe C3b as an opsonin molecule. Now, what does an opsonin molecule do? Well, it participates in a process known as opsonization, and that's not very helpful, because of course that if you don't know what opsonization means, well, if you don't know what opsonin means, you're unlikely to know what opsonization means. So let me describe what opsonization is. So this is a little bit difficult because we haven't yet discussed the process of phagocytosis, but let me just give you a brief introduction to the process that we will study in a lot more detail later on. So we have discussed that as part of the acute inflammatory response, you will recruit in a lot of leukocytes. Specifically, neutrophils and macrophages are the ones that we've seen so far. Now, these leukocytes, neutrophils and macrophages, they are known as phagocytes. So both of them are described as phagocytes. Now, phagocytes are cells which are capable of engulfing, i.e. eating effectively, pathogens and other cells. They can actually phagocytose cells of the body. They're capable of engulfing cells, but in particular pathogenic cells, and then breaking them down, digesting them down. And that process of engulfing cells and breaking them down is known as phagocytosis, and this is one of the it's the major reason that we're bringing in so many neutrophils and macrophages, because we want them to phagocytose pathogens. Now, how do they recognize pathogens? Well, we've already discussed how you can recognize pathogens. You can recognize pathogens by being covered in pattern recognition receptors for pathogen-associated molecular patterns, PAMPs. And indeed, that's one of the major mechanisms by which macrophages and neutrophils will recognize pathogens and know, therefore, which cells to actually phagocytose. But another way that you can do it is by coating the pathogen in C3b, and this is an absolute giveaway sign for a macrophage or a neutrophil, a phagocyte, to phagocytose that pathogen. An opsonin is a molecule which promotes the phagocytosis of the pathogen that it's bound to, basically, and the process of promoting phagocytosis of what you're bound to is known as opsonization. Okay, so it's often compared to making this taste beautiful. It's like we're coating the pathogen in sugar, and we hugely increase the probability that the phagocyte, which is moving past this, will actually phagocytose this. So if you imagine that the phagocyte has a certain probability when it comes close to a pathogen of phagocytosing it, then that probability is hugely raised by having the pathogen coated in C3b. So that's what we mean when we say that C3b is an opsonin. The phagocytes have a receptor for C3b on their surface, and when they see C3b coating a pathogen, they instantly want to phagocytose that pathogen. It creates a huge intracellular cascade which is going to drive the phagocytosis of the pathogen. So it promotes phagocytosis of the pathogen. So it's not good for a pathogen to end up coated in C3b. In addition, before we go any further, because C3b can do other things to help kill the pathogen besides opsonization, I also just want to mention what C3a does, because C3a, this small fragment of C3, actually does something very important. So C3a is going to potentiate the acute inflammatory response. It's going to maintain it. So C3a is what is known as an anaphylotoxin, and we'll see another molecule, namely C5a, which is also given this great, terrifying sounding title, anaphylotoxin. And these are capable of activating mast cells to release more histamine. And indeed, if you like, this is a propagation of the inflammatory response. You see, if the complement pathways are active, if you're getting C3a produced, what does that illustrate to you? That illustrates that there are still pathogens present in the tissue. If there weren't pathogens present in the tissue, the complement cascades would not be active and you wouldn't be getting more C3a produced. So having C3a continually being produced shows you that there must still be pathogens present in the tissue. So what is C3a going to do? It's going to activate sentinel cells to continue producing molecules, the pro-inflammatory mediators, basically, to maintain the inflammatory response. So C3a then, what it's going to do is it's going to go and bind to receptors specifically on mast cells. So I'll draw my mast cell here. And the mast cell will have a receptor known as the C3a receptor on its surface, which I'll just draw as this blob here. So the C3aR is meant to represent the C3a receptor. And what will happen is the C3A molecule, shown again in green here, will bind to the C3A receptor on the surface of the mast cell. So here's the C3A receptor in blue here, and that will activate the mast cell to release more histamine and also the other pro-inflammatory mediators as well. So it activates the continued release of the pro-inflammatory mediators and therefore is involved in maintaining the inflammatory response. And why? I'll go over this again. Why do you want to maintain the inflammatory response if C3A is being produced? Well, C3A being produced shows you that the pathogen is still present. Even if the sentinel cells might not be able to see the pathogen anymore, if C3A is being produced in the vicinity of these sentinel cells, then it's definitely showing that the pathogen is still present, because you wouldn't be producing C3A from C3 unless the complement cascades were activated, and the complement cascades will only be activated if there are pathogens present. Okay, so that's C3A, which will be maintaining the inflammatory response. I'll just move this down a bit to try and get the colours better. Okay, right. Next, what do I want to discuss? Next what I want to discuss is the other way that C3b can kill pathogens. So we've so far seen two of the effects of the classical complement pathway. We've seen that we'll coat the pathogen in C3b, which is an opsonin, and therefore we're going to get the promoted phagocytosis of our pathogen. We've also seen that we'll produce C3a, which is an anaphylotoxin, and will promote the maintenance of the inflammatory response. There is another really important way that the classical complement pathway is capable of killing pathogens, and this is going to be a continuation of C3b. So let me now just go over the page here. Okay, right. So let's again draw our pathogen here, and I think I'll do it this way up this time. So this is the pathogen membrane, and this is the pathogen cytoplasm here in red, and I'm just showing a portion of it rather than the entire pathogen now. And let's say that we have a C3b protein bound here on the surface of the pathogen. So this is C3b, and what colour did I previously show C3b in? I think it was in green. So we've discussed that C3b proteins will end up coating the cell membrane of the pathogen. Now, C3b proteins can actually also bind to the complex that we already had created. They can bind to the C4b, C2b complex to form now a complex of 3 proteins. So I'll draw this here. So like so. So here is C4b, here is C2b, and now C3b has joined. So let me just remind myself of the colours that I had. So C4b was being shown in blue and C2b was in red. So here is C4b in blue and here is C2b in red. Okay, so we've now got a bigger complex, and hopefully you can guess what the name for this is. This is going to be the C4b, C2b, C3b complex now. So you're going to get C4b, C2b, C3b complexes formed on the surface of the pathogen. And now this complex gains a new enzymatic activity. So previously the C4b, C2b complex was a C3 convertase. Now that we've got this C4b, C2b, C3b complex, this has the enzymic activity of converting C5 into the active form. So this is what is known as a C5 convertase. So it's going to activate complement protein C5. Right, so now let's see the next complement protein. So C5, so we've seen C1, C2, C3, C4, and now seeing C5. So I'll draw this here. So we'll have C5, I think, in yellow. So here it is, C5. And this complex is now going to convert C5, which is of course inactive. It's just the inactive form that was circulating in the blood and which has come in the inflammatory exudate. C5 is now going to be converted into a large fragment here and then a small fragment here. And the large fragment is of course called C5b and the small fragment is of course called C5a. And again, there's no confusion here. The old name is exactly the same as the new name. The old scientists did call the small fragment C5a and they called this big fragment C5b. Okay, so C5a is another anaphylotoxin. it acts on the C5a receptor on the surface of mast cells and will also cause the release of pro-inflammatory mediators, particularly histamine, and therefore it will maintain the inflammatory response. And the exact same explanation as to why we would want to do so in response to the production of C5a applies as did with C3a, i.e. this will only be produced if the pathogen is still present. If the pathogen is still present, we want to maintain the acute inflammatory response. Okay, now let's talk about what C5b is going to do. So C5b, as with all the other big fragments, is going to bind to membranes. So here is C5b, and once it has bound to the membrane of the pathogen in its vicinity, it's going to recruit loads of other complement proteins to bind with it, and these are actually going to form something known as the membrane attack complex, which will actually form a hole in the surface of the pathogen. So let me just draw this out. Okay, so I'll just give myself a little bit more membrane to work with here. So here's the pathogen continuing on. So now what's going to happen is once you've got C5b proteins on the surface of the pathogen, C5b is going to recruit other complement proteins. So the first one that will happen to bind to C5b is going to be C6 here. So C6 is now going to bind, and we'll have C6 in red, like so. And C6, it's not going to be split into two fragments in the way that the C2, C3, C4 and C5 were. So like C1, where it's just a case of it binds to something and then it's activated, C6 is like that. So C5b binds C6, then C6 is going to bind C7, then C7 is going to bind C8, and now what's going to happen is C8 is going to bind to loads of C9 molecules. Specifically about 10 to 16 C9 molecules are going to all bind in here. So I'll just show this like so. And they're going to polymerize together to make effectively a pore through the membrane, like so. So these are all C9 proteins. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's a decent number to have there. It's generally between 10 to 16 that polymerize together to form a pore. So 10 to 16 C9. So let me now just colour this in. Okay, so we'll colour in C7 then in pink, and we'll colour in C8 in yellow, running out of colours so we'll just have to reuse them. And we'll have C9 in blue here. So we've got, I think I put in 11, was it? Or 12 maybe, C9 proteins here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I think I've lost count. I think I might have double counted at one point, but it's around 11 or 12 that I've put in there, and it can vary between 10 to 16. Okay, and the principle is that each one of these little sections is a C9 protein. They polymerize together once they've been able to bind to the C8 there, or this whole complex of C5b, C6, C7, C8, and they've formed a pore in the membrane. And this just is exactly like those defensins that we discussed ages ago. Once you've got a pore in the membrane, you destroy the integrity of the membrane. Water molecules can go from the extracellular fluid into the cytoplasm of the cell, and those will then cause the cytoplasm to swell and then burst the membrane. Osmotic lysis will occur. Okay, so this structure here that's actually going to kill the pathogen, this entire thing is known as the membrane attack complex, or the MAC for short. So this is the membrane, that's the A, that's the M, attack, that's the A, and then complex, that's the C. Okay, so that entire thing that you form once you've got C5b proteins attached to the membrane is known as a membrane attack complex, and that's another way in which the classical complement pathway can help kill pathogens. So overall, we've now seen the 3 different effects of activating the classical complement pathway. Firstly, you coat the pathogens in C3b, which functions as an opsonin and gets them phagocytosed. Then you also produce the C3a and the C5a, which are anaphytotoxins and which will provide maintenance of the inflammatory response. you also finally produce these membrane attack complexes in the membrane of the pathogen, which will destroy membrane integrity and therefore promote osmotic lysis in the pathogen, i.e. killing the pathogen directly. And those are the three major effect mechanisms of the classical complement pathway. So that was the classical complement pathway of activation, and we saw that the fundamental thing which activates it is having immune complexes on the surface of the pathogen. So it did require the adaptive immune response to have occurred before it could actually be activated. The other two pathways that we're going to discuss in the next video, the alternative pathway and then the lectin pathway, those are going to be activated without the need for having the adaptive immune response occurred. Don't worry, it won't take us as long to discuss those two pathways as it did for the classical pathway, because they're extremely similar to the classical pathway, so a lot of the work for them is already done. So we'll have a break here, and in the next video we'll discuss the alternative pathway and then the lectin pathway.